Welcome. Today we explore one of the most intricate events in cell biology, prophase 1, the opening act of meiosis. Unlike the prophase of mitosis, this phase is remarkably long and complex. In fact, it is subdivided into 5 distinct stages, each with its own chromosomal choreography: leptotene, zygotene, pachytene, diplotene, and diakinesis. Let's walk through them one by one. After each section, we will show you exact NCERT lines so that it gets covered as well. The story begins at leptotene, a name derived from the Greek for thin threads. In this stage, chromosomes gradually become visible for the first time under a light microscope. They appear as long, thin, loosely coiled threads. Compaction, the gradual condensation and shortening of chromosomes, begins here and continues throughout this stage. Think of it as the cell slowly winding up long strands of DNA, preparing them for the complex events ahead. Leptotene is relatively brief, but it marks the definitive entry into meiotic prophase. Next comes zygotene, meaning paired threads. Here, something remarkable happens. Chromosomes begin actively seeking out their partners. This pairing process is called synapsis. Each chromosome aligns precisely with its homologous partner, its corresponding chromosome inherited from the other parent. Electron microscopy reveals that synapsis is not random. It is guided by a protein scaffold called the synaptonemal complex, a zipper-like structure that holds the two homologous chromosomes together along their entire length. The result of this pairing is a structure called a bivalent, also known as a tetrad, since it consists of 4 chromatids in total. These structures will become far more prominent in the next stage. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex. The complex formed by a pair of synapsed homologous chromosomes is called a bivalent or a tetrad. However, these are more clearly visible at the next stage. The first 2 stages of prophase 1 are relatively short-lived compared to the next stage, that is Pachytene. Pachytene is the longest of the 5 stages. And for good reason. This is where the genetic magic happens. The 4 chromatids of each bivalent are now distinct and clearly visible as tetrads. But the defining feature of Pachytene is the appearance of recombination nodules, molecular complexes that mark the precise sites where crossing over will occur. Crossing over is the exchange of genetic material between non-sister chromatids of homologous chromosomes. It is not random breakage, it is an enzyme-mediated process carried out by an enzyme called recombinase. This enzyme orchestrates the physical exchange of DNA segments between the 2 chromosomes. The outcome is genetic recombination. Each chromosome now carries a unique combination of alleles from both parents. By the end of Pachytene, recombination is complete, but the chromosomes remain physically linked at the sites where crossing over occurred. NCERT states that during this stage, bivalent chromosomes now clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between 2 homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over leads to recombination of genetic material on the 2 chromosomes. Recombination between homologous chromosomes is completed by the end of Pachytene, leaving the chromosomes linked at the sites of crossing over. Diplotene begins with an important dissolution. The synaptonemal complex breaks down. With its molecular glue removed, the 2 homologous chromosomes of each bivalent begin to repel one another. They try to move apart, but they cannot fully separate. They remain connected only at the specific sites where crossing over occurred. These X-shaped connections are called chiasmata. singular, chiasma, and they are the visible record of every recombination event that took place during Pachytene. Diplotene is notable for another reason. In the oocytes of some vertebrates, including humans, this stage can last for months or even years while the egg cell waits to complete development. NCERT states that the beginning of diplotene is recognized by the dissolution of the synaptonymal complex and the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the sites of crossovers. These egg-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotene can last for months or years. The final act of prophase 1 is diakinesis, the transition stage that bridges prophase and metaphase. Diakinesis is characterized by terminalization of chiasmata. The X-shaped connections move towards the ends of the chromosomes as the bivalents continue to condense. By this point, chromosomes are fully condensed, compact, and well defined. Simultaneously, the meiotic spindle begins to assemble, preparing the apparatus that will physically pull the homologous chromosomes apart. Two key nuclear landmarks disappear. The nucleolus fades away, and the nuclear envelope breaks down completely. With the nuclear envelope gone, the cell is ready. Diakinesis is the gateway to metaphase 1, and the chromosome pairs, the bivalents, are primed for separation. NCERT states that the final stage of meiotic prophase 1 is diakinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents transition to metaphase,